resting membrane potential now. So resting membrane potential means the voltage difference across the cell membrane in resting condition. Alright. It exists in all cells, not only in the neural cell, but it exists in all of the cells of the human body. Generally, the standard value of the resting membrane potential is minus 70 millivolts to minus 90 millivolts. Alright. So this you have to remember, the general value of the resting membrane potential. Now let us move on towards the basic concept of this resting membrane potential. Initially, we have sodium potassium ATPase pumps located in each and every cell of the human body. They are responsible for transferring 3 sodiums outwards of the cell membrane and passing 2 potassiums inwards of the cell membrane. That is, they help in maintaining the concentration gradient of sodium and potassium ions. They use ATP energy for doing this job, right? So this is clear that each and every cell of the human body has sodium potassium ATPase pumps. So what they do, they transfer 3 molecules of sodium outwards and 2 molecules of potassium inwards. As a result, the concentration of sodium ions outside of the membrane increases, and the concentration of potassium ion increases on the inside of the membrane. This results in creating a net negative charge towards the membrane as compared to the outside of the membrane, which is very less. If we'll see, just because of the sodium potassium pump, initially say if it was 0 millivolts, then because of this pump it will get to minus 5 millivolts, but still this is not the resting membrane potential which we'll need. So now we come to the leaky potassium ion channels, which are also present in the membrane. They do not use any kind of ATP energy, but they keep on passing the potassium ions outwards of the cell because of the concentration gradient, because we have already seen that the potassium ion inside the cell is very high as compared to the outside. Alright, so the potassium ion keep on moving towards the outside, which results in creating the more negativity on the inside of the membrane because the positive charge is flowing outwards. So these are the leaky potassium ion channels which are responsible for moving the potassium ions outwards, which result in creating this negative potential, more negative, and it takes it to minus 90 millivolt. Alright. Then we have another kind of channels which are called leaky sodium ions channels, which are responsible for again transferring sodium along their concentration gradient, that is from outside to inside. So since the positive charge now is coming inside the cell membrane, the membrane potential will drop towards more positive, that is from a more negative minus 90 millivolt it will turn into minus 70 millivolt, which is the general resting membrane potential. And let me be very clear with this, that the permeability of potassium ions throughout the cell membrane in resting state is much larger as compared to the sodium ions. And besides this, the inside of the cell also has certain anions, for example the phosphate ions and the protein anions, which are responsible for maintaining the negative potential inside of the cell membrane. So this was the basic concept of the resting membrane potential. Now we'll see the Nernst equation, which is used in calculating the membrane potentials. We just need to remember the equation and remember the values. Will not be asked to calculate any values. We have to just remember that the resting membrane potential is generally minus 70 millivolts. So the Nernst equation says that the membrane potential of any ion can be calculated by 61.5 into log base of 10 of the concentration of that ion on the outside of the membrane and the concentration divided by the concentration of that ion on the inside of the membrane. So we do not have to worry about this. This is just the basic concept of calculating how the membrane potentials are calculated. Alright. So now let us move on towards the graded potential. This is the most important topic. Now since the cell membrane is not always resting, it has to work. So first of all, the resting membrane potential can be moved towards a more kind of less negative value or more positive values, so that the cell becomes excited. This is called the threshold potential. That is the moving of the membrane potential from minus 70 millivolts to minus 55 millivolts, which is the threshold potential. At this threshold potential, all the sodium ion voltage gated channels get open, and the cell is moves on to get depolarized. So this is, we are moving more or less towards a depolarization, and hence it is said excited postsynaptic potential. All right, excitatory postsynaptic potential. We have already seen about TPSPs and IPSPs in the previous lesson. And the other situation is the resting membrane potential minus 70 millivolt can go towards a more negative value, which is minus 90 millivolt, and this situation is called hyperpolarization. Since it has nothing related to nothing to do with the excitation of the cell or the transferring of the action potential or depolarization, hence it is called inhibitory postsynaptic potential. All right. So now let us see the mechanism behind the excitatory postsynaptic potential and inhibitory postsynaptic potential, EPSPs and IPSPs. So these are the ligand-gated ion channels located on the membrane of the neurons. These ligand-gated ion channels, say for example, first of all, they are stimulated by a excitatory neurotransmitter, which is for example glutamate. There are a lot of other excitatory neurotransmitters as well. So what they do is they open the ligand-gated ion channels, and the positively charged ions, say the sodium ion and the calcium ions, start entering the cell membrane because of these ligand-gated ion channels. So the membrane potential now from minus 70 millivolts turns towards a less negative value, which is minus 55 millivolt, because of these positively charged ions. So this is the state of excitation, and hence it is called excited postsynaptic potential. All right. Then the next situation is if these ligand-gated ion channels are stimulated by a inhibitory neurotransmitter, for example GABA, gamma aminobutyric acid, then the negatively charged ions start entering the cell through these ligand-gated ion channels, and the potassium ion which was present inside start moving towards the outside. As a result, the inside of the cell membrane starts to become more of negative. So this is the situation in which the membrane potential becomes more negative from a minus 70 millivolt to a minus 90 millivolt. All right, and this is obviously called the inhibitory postsynaptic potential. The EPSPs are always greater than IPSPs. This is the basic graph through which we can see that only if the EPSPs will reach towards the threshold potential, then only the cell will go towards the state of depolarization. Before that, it will not be in a state of depolarization, which is called the all or none phenomenon, all or none principle. Let me write this: all or none principle. Right.